So in this video, we are going to discuss triple sugar iron auger, or the TSI slant. TSIs come to us in test tubes, not petri dishes like the rest of the media that we work with. Here you can see an uninoculated TSI. They're kind of red in color, and they have a distinctive slant portion and then a butt portion. So you've got two different parts to this one tube. And when you inoculate a TSI, you take your loop, you stab all the way to the bottom of the tube, and then you do a fishtail up the slant. So TSIs are very differential, but they are not selective at all. Everything can grow on a TSI slant, but they are differential based on three different qualities. The first quality that we're going to talk about is sugar fermentation, as sugar fermentation is something that we often look at with gram-negative bacteria. TSIs, or triple sugar iron, there are three different sugars in this auger. You have glucose, lactose, and sucrose. Those are the three different sugars. Some organisms can ferment all three, some can only ferment one or two. So the way that the organisms grow on the auger and the color that it turns tells you which, if any, of the three the organisms are able to ferment. If the organisms are only able to ferment glucose and they're not able to ferment any of the other sugars, they will turn the TSI slant red and the butt will be yellow. So you get this two-toned color, pink red on the slant, and then yellow in the butt. What's happening here is there is actually a lot less glucose in a TSI than there is of the other two sugars. So organisms that are able to ferment glucose will start utilizing the glucose in the auger, and they will actually turn the whole entire auger yellow. But then they run out of glucose. And if they can't use the lactose or the sucrose, they will start to use the other components of the media, and the media will turn back to the red color it originally was, which is why you get the red up here on the slant and then the yellow in the butt. If given enough time, the whole thing will eventually turn red again, but that takes a lot of time. If the organisms are able to ferment glucose and at least one of the other sugars, then the whole entire slant will be yellow. You'll have a yellow slant and you'll have a yellow butt. This is what we see for a lot of those lactose fermenters that we were already growing on media like mac and stuff like that. In this case, they utilize all that glucose first because glucose is very easy to break down, but then they switch over to those other sugars, the lactose and or the sucrose, depending on the organism. So because of that, they produce lots and lots of acid, turn the whole entire tube yellow. Another thing that triple sugar iron auger is good for is looking for hydrogen sulfide gas production. Like the hectone plates that we work with, TSIs contain sodium thiosulfate and ferric citrate. Sodium thiosulfate can work as a sulfur source if organisms are able to produce hydrogen sulfide gas, and then ferric citrate can be an indicator. If the organisms are able to break down sodium thiosulfate and create that H2S gas, they will produce a very dark black pigment in the auger. Here you can see a TSI that has a pink slant and a pretty much black butt, but you can see a little bit of yellow if you look in here. So you can still assess sugar fermentation even when you have hydrogen sulfide gas production. The H2S is going to turn it black, but you can still see the colors of the slant and the butt. So looking at this TSI slant, you can tell that this organism definitely produces hydrogen sulfide gas, but it also is only able to ferment glucose because you get that red slant and then yellow in the butt. Here you can see another organism that's able to produce H2S, as you can see down here in the bottom, but this organism is able to ferment glucose and at least one of those other sugars. And you can tell that because the slant and the butt are both yellow. The last thing that the TSIs are able to tell you is how the organisms live in terms of oxygen. Because you have the slant being a very oxygenated environment and the butt being an area with less oxygen, organisms that are facultative anaerobes can grow throughout, whereas strict aerobes are only able to grow on the slant. We do have organisms like this in our class, and you can always tell these organisms because you'll see growth on the slant and you'll get some kind of a color change. And then in the bottom, you will get no color change at all. So here you can see an organism that is growing on the slant. You can see the growth on the slant, and you can also tell that there's a more pink color than the red. But then the bottom looks exactly like the uninoculated tube, showing that there was actually no growth in the bottom of the tube.